Hi, everyone. My name's Emily May. I'm the Agricultural Conservation Lead with the Pesticide Program here at Xerces. I work with farmers, land managers, researchers, and communities to reduce pesticide risks and support pollinator conservation across different landscapes. Today, I'm going to be talking about monarchs, not just their incredible migration, the importance of milkweed, but the less visible threat of pesticide exposure. So we're going to talk about the risks of pesticides to monarchs. First half of the talk might be a little bit doom and gloom, but I promise there are solutions that we're going to talk about towards the end of the presentation, what we can do to create high quality habitat for them across all kinds of settings. I think this talk is more geared in a way towards residential landscapes than farm landscapes, but we'll talk a bit about both. All right. So every year across North America, monarch butterflies make one of the most extraordinary migrations in the insect world. They are traveling thousands of miles across a patchwork of landscapes, and it takes multiple generations to complete this journey. In the eastern population, monarchs move north through the spring and summer, laying eggs along the way. And then in late summer, a special generation, one that doesn't reproduce right away, but instead starts this long flight to overwinter in Mexico. Then there's a Western population. There are milkweed and monarchs breeding in nearly all parts of the West from June to August. The butterflies migrate from inland breeding areas to groves across along the Pacific coast in California down to Baja, Mexico, clustering in coastal groves of trees to survive the winter. It's probably all familiar with this if you're on this webinar, but let's take a quick look at the monarch life cycle, starting with the egg stage, which lasts about 3 to 5 days. Eggs are laid on milkweed, which is the only host plant that monarchs use. There's species of milkweed in North America. Next is the larval stage, the caterpillars, which last 10 to 14 days. During this time, caterpillars are constantly feeding and molting 5 times as they grow. Then comes the chrysalis stage, the pupal stage, which lasts several days to a couple of weeks. This is when metamorphosis into an adult happens. And then finally, the adult emerges. Most breeding generation adults are living 2 to 5 weeks, but the migratory to overwintering generation can live 6 to 9 months, long enough to start the cycle again in spring. The entire cycle from egg to adult can take a month or so or more, and there are multiple generations each year. So monarchs are interacting with the landscape really all season long. Eastern monarch populations have declined significantly, nearly 90% since the 1990s, and Western populations in California have dropped even more sharply. Long-term surveys at the Mexican overwintering sites show an annual decline of about 9% a year for the last 22 years. Unfortunately, this means that there's a decent chance that the Eastern population reaches quasi-extinction levels, which is a level at which the population is too low to recover in the next 10 or 20 years. And there was an updated estimate from USGS last year that estimated that risk at about 29% in the next 10 years and about 52% in the next 20 years. There's still quite a wide range in those predictions. There's still room to shift this trend. But it's significant enough that, of course, there are many of us concerned about how we can conserve this imperiled species. There are lots of drivers involved in this decline. Habitat loss is really a big one, both in the breeding and migratory range and at the overwintering sites, both in Mexico and California, as well as climate change and pesticide use. Those are kind of the three big drivers: habitat, climate, and pesticides. Those drivers of decline have shifted over time. Before the 1990s, the primary driver was habitat loss, forest loss in Mexico, as well as loss of prairie habitat in the Midwest. In the 1990s to the early 2000s, driven largely by milkweed loss in that Midwestern area. We lost over 1 billion stems of milkweed as herbicide-resistant crops became the dominant form of production in the Midwest. And then after the early 2000s, a more recent analysis showed that climate and then county level pesticide use, especially neonicotinoid insecticide use, appear to be some of the main drivers over the last 20 years. All the challenges that monarchs face interact with each other. This is true for all animals. All of those things are important to address as we try to help this species avoid extinction. But today, I really want to just focus on pesticide risks to monarchs. And we can talk about many of those other struggles at the same time, but I want to go into the details here. Monarchs are exposed to complex mixtures of pesticides across their life cycle and often at every life stage. Pesticide residues show up pretty much everywhere we sample for them, whether in ag fields and backyards and wetlands, deep into public lands. Monarchs aren't being exposed to a single product or a single dose. They're facing complex mixtures, and these mixtures can have unpredictable effects in combination. Pesticide use is widespread. This is a map here of just one chemical, clothianidin, which is a neonicotinoid insecticide that's commonly used as a coating on seeds planted out as row crops. Again, just one insecticide. But many pesticides are applied across hundreds of millions of acres, not just to crops, but also in backyards, in municipal parks, even natural areas. Those residues can end up contaminating plant leaves, nectar, and other areas. Over my lifetime, the overall volume or amount of pesticides being applied, the amount of insecticides being applied to crops has declined. But the total toxicity to insects, at least as measured as toxicity to bees, has more than tripled over that time frame. So this range-wide use of pesticides poses a challenge to building safer spaces for monarchs. So how do we understand pesticide exposure? We need to understand the pathways by which monarchs and other insects might be exposed to a pesticide. Adult monarchs are moving around, flying, sipping on nectar from flowers and laying eggs. When they're moving through an area, they might be sprayed directly, or they might pick up contaminated nectar from a previous application. But adults have this benefit of being quite mobile. They can move around from patch to patch. Maybe they forage on one contaminated plant, but then they move on to another one. The caterpillars, on the other hand, don't have that ability, right? They can't move out of a contaminated area if they're in one. So they might be picking up residues on the leaf surfaces of a milkweed or in the actual leaf tissue. Some pesticides are what we call systemic. They dissolve in water and they can be transported throughout a plant into its leaves and into its nectar or pollen. So, because larvae are basically stuck in place until they've finished development, maybe they move around a little bit, but they're not too mobile. They can be chronically exposed, long-term exposed to pesticides that are in and on the leaves of milkweeds that they're living and feeding on. And so, for this reason, we want to make sure that breeding habitat that host plants are adequately protected from getting contaminated with pesticides. To make sure that you and I are on the same page with terms, the word risk has kind of a specific meaning when it comes to pesticides. There's two main things that factor into the risk of a pesticide, whether that's to a human or to a butterfly. The first part is toxicity, or the capacity of that chemical to cause harm. How toxic is it? In what way is it toxic? How does it work inside a body? The other piece of that equation is exposure. So what's what we just talked about. How much of the pesticide is a pollinator exposed to? Was it exposed on the outside of its body? Did it eat the leaf tissue containing that pesticide? Or was it exposed for a long time? Basically, all of this boils down to the dose makes the poison. So if you have a very highly toxic pesticide that a pollinator is minimally exposed to, it might be less risky than a less toxic pesticide that a pollinator is exposed to constantly or over a long time frame. The most risky, of course, is a highly toxic pesticide with a high dose exposure. So this risk equation helps us start to think about how we manage risk. How can we limit risk? How can we maybe seek out a different way to manage a pest or otherwise limit the harm or the exposure? How do we know anything about how toxic a pesticide is to insects? Before a pesticide can be registered, pesticide manufacturers are required to submit to the US Environmental Protection Agency, the EPA, the results of a variety of different tests that are looking at the toxicity of that pesticide, the birds, mammals, fish, and insects, depending on the expected use patterns. And the way that EPA then assesses the toxicity of pesticides to insects, the main way, is based on standard laboratory based testing of adult honeybees. And honeybees are the model organism for all other terrestrial insects. So, in that test, a pesticide is applied at different doses to the top of the thorax of adult honeybees, just like in this picture made for you. And they look for the dose that kills half of the tested group of adult honeybees over the next 48 hours. The smaller the dose that kills 50% of those adult bees, the more toxic that pesticide is. And that is a standard dose that's called the lethal dose, or the LD50. There's many pesticides that have toxicity to bees and other insects like monarchs that isn't captured by this standard acute toxicity test. Larval insects, native bees, other kinds of insects like butterflies might be more or less sensitive than adult honeybees to different kinds of pesticides. So it's kind of a crude screen to look at toxicity to insects. There are only a few chemicals that have been tested for their specific toxicity to monarch butterflies. Eggs and larvae tend to be more sensitive than adults from that limited toxicity data that we do have. And it appears that monarchs tend to be slightly less sensitive than the honeybee to the widely used neonicotinoid insecticides. But monarchs are extremely sensitive to chlorantraniliprole. I don't expect you to repeat that back to me, but it's a diamide insecticide that has more than 100 uses in agriculture and in residential settings. It's used as a seed treatment, just like the neonics are on row crops. And now it's among the most popular insecticides for lawn grubs. It's found in lots of different lawn treatments. It's considered somewhat lower risk for bees, but it's high risk for monarchs. The other that we know to be highly toxic to monarchs are pyrethroid insecticides, which are the most commonly used insecticides for backyard mosquito treatments. They're also really common for ants and nuisance pest sprays around homes. So many important effects of pesticides are missed when toxicity tests are focusing only on adults and only on short-term exposure. And we know this from research that was done by University of Maryland professor Niranjana Krishnan, who looked at the toxicity of a few different pesticides to monarch caterpillars at different life stages. And she identified this really important secondary mode of action. So mode of action is basically the way that a pesticide causes harm. So she identified this unexpected mode of action for neonics and some other pesticides in butterflies, which is a disruption in molting at the last stage of larval development as they are trying to pupate. So this photo at bottom right is showing what that looks like in a monarch. This is called arrested ecdysis, where the larva basically can't successfully make it into a chrysalis. They get stuck. So this is what happened with the neonics. There were no other symptoms observed prior to this death, but this was the leading cause of death for those larvae moving into the pupal stage that were treated with imidacloprid at a certain dose. So this is just telling us that we need more testing. We need more data to understand how insects are responding to exposure to these pesticides. And this is why also toxicity studies that look at multiple life stages are really important. Pesticide exposure can have a variety of subtle harmful effects that might not kill an insect during that lab test, but could affect population growth over time. So some of these are hard things to characterize, like let's say carryover effects of pesticide exposure from one generation to the next, something we've seen in some bee studies. That's hard to do with a migratory species like the monarch. But to date, we don't have regulations that address a lot of these subtle risks. Some of the examples we've seen for monarchs include things like reduced body size, reduced larval growth, smaller wing length, and reduced longevity. There was a study showing that larvae exposed to a couple of common fungicides had smaller wings as adults, and that's a feature that could be really important when these butterflies need to travel long distances. It might affect something like migratory success. So this kind of is a broader point, which is really that pesticide risk is often underestimated when we're just using a very limited amount of data. It's hard to capture all of that real world risk of pesticides to monarchs. And what's required for a pesticide to be registered isn't enough information to assure safety. There's very limited toxicity data for butterflies, and there's limited to no data on chemical mixture impacts or the impacts of formulated products and inert ingredients. EPA is evaluating these chemicals one by one, but monarchs aren't just encountering a single chemical one time. They're exposed to tank mixes, overlapping applications, residues in soils and vegetation, lots of combinations. So it'd be like being exposed to something at home and at work and at school, but that risk is being assessed only one room at a time or one place at a time. So, what this ultimately means is that just following a pesticide label isn't enough to protect a monarch from pesticide use. Obviously, we should always read and follow pesticide labels very carefully, but we also need to do more than that to be able to protect monarchs and other imperiled species from pesticide risks. So let's shift into talking about habitat and how it gets contaminated so that we can understand what we need to do to create habitat that's more protected. Simply put, pesticides don't tend to stay where they're applied. Field research tells us that there's pretty widespread contamination of flowering plants near agricultural fields that receive pesticide applications, including the use of treated seeds. Some ways that pesticides get applied lead to more drift and more offsite contamination, like aerial applications by a fixed wing airplane or by helicopter. Those can deliver pretty high rates of chemicals far from where they're applied off the field. Ground-based applications move less far, but there can still be pretty high rates of pesticide drift at the field edge. So after reading lots and lots of field research papers, spending a lot of time modeling drift, the main takeaway I have is that I wouldn't want to put my pollinator habitat downwind of aerial insecticide applications or right next to tractor mounted applications. So this is the reason that we are very cautious about placing habitat in crop fields, right next to crop fields. There are farms where those kinds of infield plantings are good fit. We've worked with plenty of farms where that's the case based on pest management practices. And then there's some where we wouldn't put flowering plants in the middle of crop fields based on the typical management in that field. And instead, we would be looking for a more protected area outside of the crop fields or around the farmhouse. Having even a small unsprayed buffer between the crop and the habitat can be useful to make sure that you're not getting the highest rate of drift deposition in your planting, but instead you would have it in your unsprayed buffer. So we can talk more about that in just a bit. The other big takeaway that I have from field research is that pesticide drift and pesticide contamination is not just a problem in agricultural areas. Xerces conducted a study looking at pesticides and milkweeds across California's Central Valley, which is, of course, a highly agricultural area, but there's also a lot of big natural areas and urban areas. So we sampled 227 milkweed plants from a mix of agricultural, urban, and natural areas. And every single one of those plants contained pesticides, some at levels known to be harmful to monarchs. Across those samples, there were 64 pesticides, including a wide variety of insecticides, fungicides, herbicides, and an additive, and an average of 9 pesticides per plant, with some plants containing as many as 25. Unfortunately, we only had toxicity data specific to monarchs for 9 of those 64 chemicals. So for the vast majority of those, we don't actually know how they specifically affect monarchs. We would have to use something like a bee LD50 to be able to assume some level of risk. That's a pretty big gap. We conducted a similar study of milkweeds coming out of retail nurseries and found similar levels of contamination. So we can talk more about that if you want in questions, but I didn't want to put another slide up on it. One thing that I've learned looking at lots of different field research papers is that one big limitation is that a lot of these studies are only testing for a small number of chemicals, often just the ones that they're looking for, that they're expecting to find. Maybe that's, a small panel looking at neonicotinoids. The reason that we detected so many is because we were looking for a broad panel. There was another study that we've just conducted that's in press looking at butterfly host plants. from two different urban areas in the West, Sacramento and Albuquerque. We were surprised to find widespread contamination of those host plants from urban parks, from backyards, with atrazine, which is an herbicide that's used in agriculture that you wouldn't be expecting to find in plants that are in an urban environment, but it was there. So if we hadn't been testing for a broad panel of pesticides, we would have missed that, which really just highlights two things for me. One, we need more testing. We need more sampling across all different kinds of landscapes. And we need to make sure we're looking at a wide range of chemicals, not just the ones that we're expecting to find. So again, pesticide contamination is not just an agricultural issue, and it's not just breeding habitat that we need to protect. Overwintering sites are particularly vulnerable as they house hundreds, potentially thousands of monarchs at a time through the winter months. So this is a video here. And what you're seeing in this video is from Pacific Grove, California, which is home to one of the largest Western monarch overwintering sites. In 2024, volunteers that were conducting regular counts at this overwintering site discovered around 200 dead and dying monarchs on a private lawn outside the sanctuary, which is about 10% of that site's population in that year. And these butterflies were exhibiting clear signs of pesticide poisoning. So 10 monarchs were collected and sent for testing, and the results were pretty striking. Each monarch contained an average of 7 different pesticides, including 3 pyrethroid insecticides, bifenthrin, cypermethrin, permethrin, which were detected at or near lethal doses. So in most of these butterflies that were collected, the combined levels exceeded the LD50 that was known for monarch caterpillars. So these findings really point to pesticide exposure as the likely cause of the mortality event. But unfortunately, we couldn't pinpoint any source for that pesticide contamination. That site is surrounded by private residences, a couple of businesses, and pyrethroids are really among the most commonly used insecticides in that kind of a setting. They're frequently applied as outdoor perimeter sprays, barrier sprays for ants, other common household nuisance pests. They can be directly applied to all kinds of different surfaces: soil, turf, foundation, patios, driveways, ornamental vegetation. California has one of the strongest pesticide reporting frameworks in the country. And despite that, most homeowner applications and a lot of landscape treatments end up either going unreported or only reported at the county level, not at the site level. So it makes it very difficult to trace this kind of a pesticide exposure incident in an urban area. So what all of this means for conservation. I will be transitioning out of doom and gloom very soon, don't worry. Pesticide exposure threatens the success of our conservation work. It threatens the continued survival of this species across its range, at overwintering sites where its clustering makes it very vulnerable, and throughout breeding and migratory habitat. So the labels, as I sort of talked through, are not protective enough on their own. To recover monarchs, we really need to be reducing harm from pesticide exposure across landscapes, in agricultural fields, in the habitat we're creating, on lawns, and through policy and regulation. So what's our conservation approach here? When it comes to pesticides, we take a precautionary approach. We're not assuming no risk. We're not assuming even minimal risk. We're assuming that there could be some pesticide use in the environment, and we are looking very carefully at that in deciding how we approach habitat creation. Critically, when we plan habitat, we need to talk about pesticides. You know, we work with all kinds of growers, and there end up being hard conversations sometimes because we don't think every space is always going to be suitable for habitat. If you're on this webinar, if you've created habitat, you know that habitat restoration takes a lot of work, it takes a lot of effort. So when we have limited resources, when we're working with just a small number of dollars, we're trying to prioritize those dollars into safer areas with higher quality habitat. But that's enough doom and gloom. So let's talk about solutions. There are lots of ways to help monarchs and to help other pollinators. It can look like habitat restoration work. So restoring and protecting breeding and migratory habitat, restoring and protecting overwintering habitat, reducing pesticide use and risk in the ways that you can control, and then in participating in community science efforts, which are super critical to understanding monarch trends, as well as in advocacy and policy efforts. So now let's talk about monarch habitat. What makes high quality monarch habitat? There's three core components, all of which you see here. We've got native milkweed, we've got nectar plants, and then we need safe habitat conditions. So monarchs are milkweed specialists. Milkweed is the only plant that monarch caterpillars eat, and it's an essential part of breeding habitat, required larval food plant and adult energy source. Native milkweeds are found throughout much of the US into southern Canada, with about 72 species across North America. The second piece here, of course, nectar plants. Adults are using nectar as fuel throughout their life. Monarchs are pretty generalists when it comes to nectar, meaning they'll use a wide range of different flowers, but there are some species that seem especially attractive or beneficial. And we have nectar plant lists for different regions available on our website at xerces.org, as well as native milkweed guides for different regions as well. And then third, the habitat needs to be safe. So that means protecting it from pesticide contamination, as well as high pathogen loads, while offering some of the basic features that monarchs also use in the landscape, things like shade and shelter, perching sites. So high quality just means a space where monarchs can thrive safely and complete every stage, of their life cycle that they can complete in that space. You already know about milkweed, so I didn't include a slide about native milkweeds, but they support a wide variety of things besides monarchs. But depending on where you're located, nectar availability in the beginning and end of migration can be pretty important for fueling adults through that part of their life cycle. The best way to achieve season-long bloom and making sure you have some plants blooming from spring through fall is just to plant a diverse variety of plants with different bloom times. So we have suggested monarch nectar plant lists for different regions of the country available at xerces.org. In hot climates, trees and shrubs can also be important for providing shade for monarchs for perching. And there's some evidence that monarchs actually prefer to lay their eggs on shaded milkweed. So you might be picturing a milkweed planting in full sun, but in hot or arid areas, you might consider creating your monarch habitat under a shade tree. Or if you already have a planting, consider how you might be able to add some structure and some shade to offer that wind protection for monarchs and also shade that might encourage them to lay more eggs. There's lots of different opportunities to create habitat. Cities and towns can provide great habitat for monarchs, whether in yards and public gardens, around buildings, and places of worship, abandoned lots. You know your town, there might be some space available to create that habitat. And it can look really lovely. There's ways to incorporate milkweeds and nectar plants that can look really great. I have a few plantings in my own town that I wander by all the time, just looking to see if there's eggs and caterpillars. So then how do we protect that habitat from the pesticide concerns that we've been talking about for a lot of this presentation? So if you are starting from scratch, let's say you don't already have habitat in the ground, the starting point is choosing locations that are more protected based on what's happening around that property. And this slide shows two of our different pollinator habitat assessment guides, one for farmlands and agricultural landscapes, and the other one for yards, gardens, and parks. These are guides that are designed to help land managers identify habitat needs at a site. So what habitat already exists there? What are the deficiencies? And how could you best support pollinators with some additional management? But what it also includes is sort of a helpful checklist of practices around pest management, things like how are pests being controlled, whether pesticides are being used. So I wanted to point it out as a place to start if you want to start asking the right questions about management practices and pesticide use. It's not a full risk assessment or anything like that, but it can be helpful to just have a reminder in front of you going, well, what's going on at this site? Now, if you're in a position to influence management, whether it's your own land, it's your own farm, or it's through some kind of partnership with the land manager, this is also an opportunity before you've planted the habitat to look at pest control practices and figure out if there's any flexibility. Could some of those management practices that are currently in place be shifted towards more non-chemical methods, prevention methods that might help avoid the high-risk pesticide use? There's things like cultural methods, physical removal that can reduce reliance on pesticides and help ensure that when you do put habitat in, that it will be safer for pollinators. But of course, in a lot of cases, you might not have full control over how your neighbors are operating or how the land around your habitat is managed. So then the question becomes, what if my neighbor or my town or whoever is applying pesticides nearby? This is really common in residential areas. You might be right up against a neighbor where pesticides are being applied in ways that might not be super obvious. We're not just talking here about visible sprays. It's a lot more obvious when you're in an agricultural area when a pesticide is being applied. But there are things like granular lawn treatments or foundation drenches for things like termites, products applied through misting systems or irrigation systems. And those kinds of applications might drift or leach or otherwise move in ways that could expose neighboring habitat areas, even when you're doing everything right on your own property. So what can you do when you don't have control over neighboring practices? Or, when your neighbor or your town is spraying, or when there might be some kind of routine pesticide use nearby? There are a few solutions. So one of those is spatial buffers. So again, if you're thinking about siting habitat, physically siting habitat farther away from areas where you know pesticides are being used or are likely to be used. The farther you have the more space you have available to site it away from known areas of pesticide use, the better. Um, and the amount of space that you need to reduce harm really is going to just depend on the chemical, how it's applied, and the typical conditions there. The more pressurized or higher up that the application is made, the more likely it is to drift. By higher up, I kind of mean how high up is that chemical being released into the air. So if you're in agriculture or near agriculture, one thing you can do is check your prevailing wind direction. Try to avoid planting habitat directly downwind of fields that are receiving pesticide treatment. Likewise, slope can matter. If you can site habitat upslope of treated areas, that might help limit runoff into the habitat area. But we also recognize that space is often limited, especially on small farms or in urban areas. And this is where something like a vegetative buffer might come in. So this could look like planting dense, non-flowering shrubs or tall grasses between your pollinator garden and an area that's receiving a treatment. These plantings, an ideal candidate for a drift fence would be something like a cedar or an arborvitae or another kind of evergreen non-flowering species that's going to capture that drift as it comes in and that's appropriate for your area. The reason for using something that's not attractive is to avoid creating kind of a contaminated trap where you're attracting something into something you know is going to be contaminated. And then finally, temporal buffers. And this can look a couple of different ways, but I think the way I'm going to talk about it here is really giving time between a pesticide application and when you're putting habitat in the ground. So for example, if you are converting an area that has previously been planted with seed coatings, neonic seed coatings, or treated with persistent herbicides, it might need a year or more of fallow time before planting pollinator habitat. Just thinking about how that habitat is going to establish over time. It could also mean designing your habitat so that if you know that pesticide application happens at the same time every year, you just have stuff flowering before or after that pesticide application. None of these solutions is really perfect, but they can work even together to significantly reduce pesticide exposure and help ensure that the habitat that we worked so hard to put in the ground is actually safe and usable for monarchs and other beneficial insects. And then finally, if you're sourcing your habitat plants from nurseries, seek out pollinator safe plants, which could mean buying certified organic plants, or it can mean having a conversation with your local nursery to see how they're growing their plants. The thing you can say is, I want milkweeds or I want nectar plants that are free of pesticides that could be harmful to monarchs and other pollinators. The more that a nursery manager is hearing this from their customers, the more likely it is that they'll pay attention. And I could give a whole other webinar about this, and so could my colleague Sharon Salvaggio, but we have more information available on pollinator friendly nursery management and buying bee safe plants on our website. So I would encourage you to check it out. There are also previous webinars that focus really on how do you have that conversation with the nursery? What does it sound like, kind of gaming out the whole conversation, which some people have said has been really helpful. So those are some of the key steps for habitat protection: citing them away from pesticide use or changing practices to reduce pesticide risk, or using some combination of spatial, vegetative, or temporal buffers. Outside of habitat, another way to be engaged in monarch conservation is with community science, which is a key source of data on how monarchs and their habitat are doing. We are right in the heart of the International Monarch Monitoring Blitz this week. So if you're in the United States or Canada or Mexico, I believe, you can participate by reporting your observations of monarchs and milkweeds to your local community science project. Could be the Western Monarch Milkweed Mapper, Journey North, Monarch Larva Monitoring Project, iNaturalist. So go look it up, go look at these different programs and see how you might be able to collect and submit data. Later on in the year, if you're in California, you could participate in the Western Monarch Thanksgiving count or New Year's count. This is when dozens of volunteers visit those monarch overwintering sites in California and estimate the number of monarchs at each location. And this is really the primary way that we gauge the size of and it declines and trends in the Western monarch population. So if you're interested in that, check out westernmonarchcount.org. So a lot of the work that we do focuses on protecting habitat at that really small scale. One field edge, one pollinator garden, one planting site at a time. And all of those efforts are really important. They definitely add up. What you're doing in your yard matters. But when it comes to recovering monarch populations at scale, we do have to zoom out and look at the bigger picture. Monarchs aren't staying in just one place, they're moving across the landscape, interacting with all kinds of habitats, many of which end up having some level of contamination. So, to truly protect them, we need to be looking at pesticide risk at the landscape scale. And realistically, the only way we're going to really reduce risk is through stronger policy and more effective regulation. One place you can start locally that might be a little bit more tractable than thinking about broad policy and regulation is just asking questions about how public lands around you are being managed. Things like parks and schools, roadsides, city landscaping. Ask for information about what kinds of pesticides are being used and why and how often. There are already some communities that have adopted policies to protect pollinator habitat or pesticide free policies. So if your city or county hasn't taken those kinds of steps, you could advocate for change, something like an integrated pest management policy, prioritizing prevention, prioritizing non-chemical methods before the use of a pesticide. If you're interested in that and you don't know where to start, I would encourage you to visit the Bee City USA website for more information. They have a toolkit for what these municipal IPM plans can look like. Policy change isn't something that happens on its own. It's something that happens when there's a lot of voices pushing for it. So whether it's your school or your city, your state or beyond, your voice really can matter on this kind of an issue. So thanks for looking into it. So before I end, I just wanted to point you to the many different resources available for free on our website. We have resources on everything from monarch management, planting and establishment for native milkweeds, monarch nectar plants, broader lists of recommended plants for pollinators and beneficial insects for all regions across the United States, the guidance on nursery management and buying bee safe plants, as well as some broad guidance on how to protect habitat from pesticide contamination. And I will say we are working on updating that document currently to add a little bit more nuance on the whole spatial buffers because that really can depend on what chemical and how it's being applied. But look it up at xerces.org. Check out your regional guides. And if there's anything here that you feel like, you know what, I have this question and I don't see it answered in any Xerces resources, come tell us, because we're always developing new information and new resources to meet your needs. So I want to give a big thank you to all of our supporters, especially Xerces Society members, for their support. We can't do it without you. And the work we do really depends on everyone. So thank you so very much for sitting through this presentation. I was so worried about how much information I was going to give that I think I ended early. I cut a lot of information out thinking that I was going to talk too long. So thanks for sitting with me in all the doom and gloom. And I'm glad we have plenty of time for questions.
We definitely do. Thank you so much, Emily. That was really wonderful. You're getting some love in the chat and just gratitude for all this great information. So we're going to dive right into our questions. I'm going to start off with this first one from an anonymous attendee. If someone hires a cosmetic spray company to spray their yard, does that pesticide application render any flower sprayed poisonous to pollinators for a period of days? Example, the poison lasts after, how long does it last after the initial spraying, which I know is a really complicated question based on what they're spraying.
It definitely depends on what they're spraying. I will say that, a typical cosmetic pesticide application for, let's say, a backyard mosquito treatment that comes every couple of weeks to spray is often using pyrethroid insecticides, which we talked about a little bit. And those have a residence period of days to weeks after application. We will have another webinar in November from Aaron Anderson, my colleague, who has been working, he's the lead on our residential pesticide sampling projects. And he is in the process of publishing a paper looking at those residential mosquito treatments and drift into neighboring yards. So it really can vary depending on the application, how much is ending up in your yard from a neighbor's application.
Thanks for that, Emily. And yeah, looking forward to that webinar. All right, we have a couple people interested in, have some questions about herbicides, wondering how do herbicides factor into this? And someone else asked, I know herbicides are an issue in that they remove foraging resources from bees and butterflies, but are they also toxic to monarchs and other pollinators?
We don't know very much about direct toxicity of herbicides to monarchs specifically. The thing we know about is mostly the general indirect effect that they can have on monarch food sources, especially, milkweeds. We lost so many stems of milkweeds from the Midwest when we shifted to herbicide-based weed management in the crop fields. So I think we need more data on direct toxicity. But generally speaking, herbicides are less acutely toxic, much less acutely toxic to a monarch when sprayed or to any insect than something that's designed to kill an insect, like an insecticide. That's not to say that they couldn't have different harms, especially a formulated product that could have, a surfactant in it or something, a spreader sticker type thing that might make it hard for that monarch caterpillar to breathe if it were exposed directly to it.
All right, this next question, how can folks test their own milkweed at home if it's contaminated?
Oh, that's a great question. And I wish I had an easy answer for that. I will say part of the reason why people don't do these broad panel pesticide studies is that they're so expensive. Typically speaking, we're looking at, upwards of $100 a sample to test a single sample for pesticide residues. So if you, let's say, I'll give you one example. If you're testing for one single chemical, it can be less expensive. So if let's say that you bought a house that had a foundation treatment before you moved in, and you're trying to decide, well, should I put any kind of habitat around the outside of the foundation? You could maybe send off the soil there for testing one specific pesticide, which in the case of termite applications tends to be fipronil, and just see if you have that pesticide. That might be a lower cost way of looking for pesticides. If you're looking for a broad panel, like you testing your milkweeds, it's going to be quite a bit more expensive. If you're willing to shell that out, get in touch with us. We have a list of labs that we've worked with in the past, and could probably direct you. But it's not easy to do, single sample testing of pesticide residues, unfortunately.
Yeah, understandable, but appreciate it if you guys do, if someone does decide to go that route. All right. Bethany wants to know, my dog has a tick collar. Can the chemicals from this in his hair do damage to monarchs and other insects in the yard? They've read that baby birds can die from being in nests made from this, but they're wondering about how it can also affect monarchs.
Yeah, so that was a study that just came out in the past year. Thanks for bringing it up, that the veterinary medicines that are applied to dogs can end up in their fur. And then if you're, let's say, brushing out your dog outside and all that fur gets loose in the yard, that a bird collecting that fur to be lining their nest can actually experience lower, nesting success of their young birds. I don't know how that might affect monarchs. It seems like a less direct pathway of exposure for a monarch from a dog's hair to, a monarch caterpillar, and a more direct one for a bird that's collecting the fur to build the nest. But since that study came out, I will tell you, I've been brushing out my dog inside and putting the hair into my garbage rather than brushing him out outside.
Same. I've been doing the same thing. Something came up on the Bug Banter podcast about that. And I was like, we have a very furry dog that sheds all the time. So yeah, brushing him inside as much as we can. All right. I did want to note that Emma Pelton, who is one of our monarch specialists in our endangered species team on staff, is here on the podcast or on the webinar. And she just wanted me to mention that it's worth mentioning that herbicides may be worth the trade-off for establishing or managing pollinator habitat. There's a lot of anti-herbicide people, but for restoration, there might be that might want to bring up that herbicides are sometimes a tool in the toolbox for the greater good. But I think it's just every situation is so dependent and so different. So yeah, thank you, Emma, for bringing that up.
Yeah, absolutely. And if you're interested in that, we did a Xerces webinar on this exact thing called herbicides, a double-edged sword for restoration. So you can look that up on the Xerces YouTube channel. But absolutely, they can be a useful tool in the toolbox for restoration and invasive management.
Yeah, conservation, unfortunately, is sometimes not super straightforward. All right, this next question I want to ask is Lisa. She's actually one of our volunteers and a nursery owner. Has a question: are pyrethrins as damaging to monarchs as pyrethroids?
Oh, I'm so glad you brought up this question, because one of the slides that I had here originally was about organic pesticides, and then I ended up taking it out for time. So pyrethrins are the chemical that pyrethroids are sort of based on structurally. They're the organic version that's derived from chrysanthemum flowers. Pyrethrins are highly toxic on contact to monarchs and other insects. But they break down quite quickly in the environment. So they are a lot more unstable in the environment. The whole point of developing a synthetic version was to make them more stable and more persistent in the environment. So you can also see this in sort of advertising for mosquito treatments in backyards, where you might get advertising, from the person knocking on your door saying, hey, these chemicals are derived from chrysanthemum flowers. But there's a real distinction between the one that breaks down in a couple of days and the one that breaks down on the order of days to weeks. That's the pyrethroid. And that's typically what those backyard treatments are actually made of, is the ones that were implicated in the monarch kill in Pacific Grove, California. So I'm glad you brought that up. I think while I'm on the topic of organic pesticides, just because it's organic doesn't mean it's free of risk. On the whole, organic standards are strong and something that we support. When I talk about putting plantings in the middle of a field, a lot of times those farms are organic farms where we're putting, flowering habitat in the middle of the crop field. But there are certain organic pesticides that you should be aware of that could cause harm to a monarch caterpillar. Among those include neem oil, which is a really common pesticide used in home gardens. So neem oil, the main component of it is something called azadirachtin. It's an insect growth regulator, which is something that affects the ability of a developing insect to continue molting and successfully make it through its caterpillar life. So you wouldn't want to use neem oil on a host plant where you've got larval butterflies. The other one that I guess the two more, one would be spinosad, which is a highly toxic on contact organic insecticide. And then the third is BT. There are different strains of BT depending on what insects you might be targeting. There's the BT for mosquitoes that has become ever more popular for mosquito bucket traps for mosquito management, and that targets flies. But there are BTs that target Lepidoptera or butterflies and moths. So those formulations are highly toxic to a larval monarch butterfly. All things you could find in our guide to organic approved pesticides on the Xerces website.
Thank you, Emily. Yeah, we'll post the link to that in the chat. Next question, Karen wants to know, is the chloran pesticide tested for toxicity for monarchs or just the neonics?
Yes, so that's the reason we know about chlorantraniliprol's toxicity to monarchs is because it was included in the suite of pesticides that were tested by Niranjana Krishnan. She did bioassays on different instars of monarch caterpillars as well as adult monarchs, and found that that chemical was 50 to 500 times more toxic than neonicotinoid insecticides to monarch caterpillars. When it was released, it was heralded as being exceptionally toxic to both beetles and lepidopteran pests, so caterpillar pests. So of course, it also is quite toxic to monarch caterpillars.
All right. I have a little bit of a policy question. Michael wants to know what you think of the new New York state law that banned the use of neonic-coated seeds in agriculture. How did the bill sponsors overcome resistance from the Farm Bureau and other allies? Are other states considering similar bills?
Oh, interesting question. So the bill is the Birds and Bees Protection Act in New York State, and it phases out the use of neonicotinoid coated seeds on a few different row crops, really the primary row crops. If you don't know, neonic treated seeds are used on almost 100% of corn acreage across the United States, and they are particularly toxic to bees. I think we are generally in support of the legislation that's moved in New York and then in Vermont that are phasing out the use of these prophylactic seeds. Both of the bills in New York and Vermont include basically an exemption process, an emergency exemption process based on pest risk assessment. So there is an avenue to continue use of those seeds when it's it's not just a prophylactic use, not just a preventive use, it's actually based on whether or not the pest is posing a risk in that field. Lots and lots of field research at this point showing that those seeds are only useful on a very small acreage, especially up here in the Northeast, like 5% or less of fields ever see the amount of pest pressure that could lead to yield damage. But they're used on almost 100% of acreage anyway, causing significant downstream contamination. So yes, we're in favor. I think there's always regrettable substitutions involved with pesticides. It's like playing whack-a-mole where you manage to figure out the harm from one thing and then the next thing pops up. So I think the thing we're looking at both in New York and Vermont is what's the next thing that's going to be coming on seeds? If it's not neonicotinoids, unfortunately, it's probably diamides, which include chlorantraniliprole and similar chemicals in that same insecticide class. So we've maybe solved a problem for bees and created a slightly additional problem for caterpillars. So continuing to push for seeds and seed coatings that are being used as part of an integrated pest management program where we're scouting fields for pests and understanding when we might have a risk of a seed pest, and then only using those seed coatings when we recognize that threat and maybe not in the situations where those pests are not creating that kind of risk in the field. That was a long answer. I feel like I've been giving long answers to all of these questions, but hopefully
No,
that.
that's okay. It's really appreciated. Everyone really appreciates all of the information you're sharing. I think you touched on this in your presentation, but in case folks missed it, I think it's a relevant question anyway. If a field has been used for ag crops treated with neonics, what is the half-life of that residue in the soil? They want to plant a native seed mix in that field, and they're wondering if the perennials, wild grasses, and forbs absorb neonics in early growth. And then do they retain that toxicity through the entire life of the plant?
So great question. So when it comes to a field that has been planted with neonic treated seeds, the half-life of different neonics can be quite long. It can be months, in some cases over a year for a half-life. For the primary, so clothianidin and thiamethoxam are the primary seed treatments at this point. The way I like to look at it is if you had a treated seed crop going in May, let's say, and then grown through the season, harvested, that following season is when you would want to be doing some kind of fallow. We would also call it site prep. So you can do site prep through the next season. Maybe you want to do some, grass cover cropping through that year. Depending on what the weed pressure looks like, you could use other methods of prep. But at the end of that season, you could then seed and know that your seeds are going to come up basically 2 years after that last planting of a treated seed. So there might be some amount of residue left in the soil at that point, but that 1 year of fallow is going to go a long way to dissipation of those residues. There were a couple of studies that looked at neonic residues in prairie strips, and these are flowering strips that are planted at the foot slope or in the middle of fields. And these were prairie strips that had been planted 2 years following the last use of a treated seed, and they didn't find any neonics in those plants. So I think that 1 to 2 year fallow period is going to give you a good basis for your prairie planting.
I'm continually impressed with all of the names of chemicals that you're pronouncing.
It is like another language.
It definitely is. I'm glad no one's thrown too many in the questions because I would butcher them. Tim wants to know, can you make some recommendations about establishing pollinator habitat or, some monarch habitat in a former lawn setting? In particular, they have a person who wants to use herbicides, but does not want to use herbicides at all, but they are concerned about them being outcompeted by non-native plants.
So is the question about site prep for the planting?
Yeah, it sounds like they basically, without using herbicides, what would you recommend that they do if they're trying to get rid of their lawn and establish native habitat?
Got it. Yeah, it can depend on how much area you're working with. So I'm testing out different methods for prepping my current lawn for pollinator habitat. I had a lot of success using a sod cutter. So I rented a sod cutter from a local equipment rental company and basically cut the lawn a couple inches down, basically under its roots, and then rolled it off and then seeded into that. And there's been a little bit of competition from the previous grasses, but it's been really successful overall. The other methods I'm going to try are, it's very easy to purchase used billboard vinyl. So I am planning to do, a vinyl prep method and then a cardboard sheet mulching prep method. And then I might try one that is basically tilling in and then using buckwheat as a cover crop. A couple successive plantings of flowering buckwheat through the season to just suppress weeds, see if that works. But I will say the sod cutter has worked great for that long setting.
Thanks so much for sharing your experience there. Good luck.
Thanks.
All right. Pete wants to know, could you please discuss the 3 metabolites from the milkweed pesticide survey?
Oh, what were the metabolites? I don't know off the top of my head what the degradation products were that were in the milkweeds. But it's also, I'm going to get on my high horse again about looking at a broad suite of different products when you're doing a pesticide residue analysis. Those metabolites can be actually quite toxic themselves, depending on what it is. And sometimes a pesticide can break down from one chemical into another chemical. So I already mentioned thiamethoxam and clothianidin, the two primary neonicotinoid treatments available. Thiamethoxam breaks down into clothianidin. That makes sense. So if you are sampling plants around a field that has been treated with thiamethoxam, you should really be looking for clothianidin because it breaks down pretty quickly into clothianidin. So short answer is I don't know what those three metabolites were, but they could be important.
All right, we're going to end today on this last question from Jennifer. They're wondering what the most gentle way is to approach a neighbor about spraying pesticides. I think that is a question we get a lot, and it's a hard one to answer, but I'm gonna ask anyway.
That can be really tough, and often it comes down to just knowing your neighbor and knowing what might land with them. I think if I had a neighbor that was applying pesticides, I might go talk to them from a like kids and pets perspective, because that's what I worry about the most is, what is my kid, what's my pet being exposed to? But other people might have other entry points to that conversation. And I think it really comes down to just understanding, what is it that's leading them to use that pesticide, and validating the things that they might be using it for. It might be for mosquitoes, it might be for ticks, maybe they've experienced Lyme disease. There's all kinds of reasons why people end up choosing to apply pesticides. So coming at it from sort of a place of empathy rather than shame or accusation is good, and just talking about the things that you value and the reasons that you have questions about it. I will say then, if you can't do anything about what your neighbors are doing, the drift fencing and other things can help protect your habitat. And I will say there's if you're in a highly trafficked neighborhood, a lot of signage can be useful. There can be signage that kind of explains your perspective on pesticides and also helps keep salesmen away from your door. Like this yard is pesticide free for XYZ reasons. That might just help even prevent you from having a conversation you don't want to have.
That's really good advice. Yeah, I feel like some people just don't know that like some chemicals kill all bugs, that they think they really are just tailored to like one type of insect, and that's not normally the case. So yeah, I like the approach of empathy. I think goes a long way. Well, thank you so much, Emily. This was very informative. I learned so much, and yeah, you're getting lots of appreciation in the chat. So thank you so much for joining us today. And we also recorded Bug Banter yesterday. That will be coming out later next month. If you listen to our podcast, you can hear Emily dive into some of these topics as well. So thank you so much, everyone, for joining us. We hope you have a wonderful rest of your day. Thanks, Amber.
Thanks so much for being with us today.